Mm.
Morning, everyone. The topic of my presentation today is neurobiology of mood disorders, and my moderator is Dr. Shelly ma'am. So we'll be covering the neurobiology of mood disorders under various headings. So we'll be covering neuroanatomy, neurophysiology, neuroendocrinology, neuroimmunology, neuroimaging, and then genetics. So starting off with the neuroanatomy. So basically, there are four major brain regions which are involved in the regulation of normal emotions. So these are the prefrontal cortex, the anterior cingulate cortex, the hippocampus, and the amygdala. So now here in this figure, we'll see the various areas involved. So first of all, here we have the prefrontal cortex. So prefrontal cortex, it is basically the part of the frontal cortex apart from the primary motor cortex, the supplementary motor cortices, and the frontal IQs. So this prefrontal cortex, it is basically involved in the higher order functioning, the emotional functioning. So it has basically been divided into four parts. So first of all, we have here is the dorsolateral prefrontal cortex, which is basically involved in the executive functions, the task setting and the monitoring. Then we have the ventromedial prefrontal cortex here. This is basically involved in energization. Then the orbital prefrontal cortex, which is involved in behaviors and emotions. And also we have the frontal poles, which have not been shown in this diagram. So here we see amygdala and the hippocampus. So both of these are parts of the limbic system. So amygdala is basically considered as the functional center of the limbic system. It is important for processing of the emotional stimuli. Whereas hippocampus, so hippocampus basically, hippo means horse and campus means sea. So it resembles the shape of a seahorse, hence the name. So hippocampus is basically involved in the consolidation of memories, the formation of new memories, consolidation, and together with the amygdala, it is responsible for emotional memory. Next, we have the anterior cingulate cortex here. Now, this anterior cingulate cortex, it lies at the frontal part of the corpus callosum. Now, anterior cingulate cortex, it is involved in attention, motivation, and environmental exploration, and integrates emotional and attentional inputs. So now let us come to the neurophysiology. So first of all, we will be just going very briefly about the pathways of the biogenic monoamine neurotransmitters. So the monoamine neurotransmitters are the dopamine, norepinephrine, serotonin, acetylcholine, and histamine. So first of all, we study about the dopamine pathways. So dopamine we know is a neurotransmitter and it is formed from the tyrosine. So it has basically 5 pathways: the mesolimbic, mesocortical, nigrostriatal, the tuberoinfundibular, and the thalamic dopamine pathways. So let us now look at this figure. So this pathway here is the tuberoinfundibular pathway, that is from the infundibular hypothalamus ganglion hota hai, usse from the hypothalamus to the anterior pituitary. And this pathway is basically important for, as it inhibits the secretion of prolactin. So basically, when we get dopamine antagonists, so there is prolactin in the prolactin is raised and it leads to gynecomastia, galactorrhea, amenorrhea, sexual dysfunction. So another pathway is this one. This is the thalamic dopamine pathway. So not much is known about this pathway. So the thalamic dopamine pathway, it innervates the thalamus from multiple sites in the brain. Now look at let us look at this pathway. This is the nigrostriatal pathway from the substantia nigra to the striatum. The striatum is basically part of the basal ganglia. It is formed from the caudate nucleus and the putamen. So this pathway is basically a part of the extrapyramidal system, which we call, and is responsible for movement disorders. Next, we have 2 very major pathways: the mesolimbic and the mesocortical pathways. So let us look at this. This is the mesocortical pathway, that is, it projects from the midbrain, so ventral tegmental area in the midbrain, to the motor cortex, the ventromedial prefrontal cortex and the dorsolateral prefrontal cortex. Also, we have this pathway. This pathway is the mesolimbic pathway from the ventral tegmental area in the midbrain to the nucleus accumbens in the limbic system. So here, if this is a diagram showing the classical mesolimbic pathway. So basically, dopamine is responsible for reward, pleasure, motivation. So whenever there will be hypoactivity of this pathway, it would lead to symptoms like lack of energy, apathy, and lack of motivation. Now, next we have the classical mesocortical pathway. First to the ventromedial prefrontal cortex. Now, this pathway is responsible for the affective symptoms. And the pathway to the dorsolateral prefrontal cortex is responsible for the cognitive symptoms in mood disorders. So now, next is the norepinephrine pathway. So this here, the cell bodies of the noradrenergic neurons are located in the nucleus ceruleus here, and then they project to the various areas of the brain. So noradrenergic projection to the amygdala and the hippocampus are implicated in emotional memory and behavioral sensitization to stress. Prolonged sensitization to stress and prolonged activation of locus ceruleus contribute to the state of learned helplessness. Now, next is the serotonin pathway. Now, serotonin pathway, basically we have here in the midbrain the raphe nuclei, from where there are projections of the serotonergic neurons to whole of the brain. Now, serotonin, it basically regulates sleep, appetite, libido, body temperature, metabolism, and the circadian rhythms. There is some evidence also suggesting that the serotonin neurotransmitter is under genetic control. Polymorphism of basically the serotonin promoter region of the serotonin transporter gene has shown to moderate the risk of depression. Also, serotonin also has a seasonal influence to it. Now, next is acetylcholine. So, acetylcholine, basically, we have the nucleus basalis of Maynard, which contains a large population of cholinergic neurons that project their axons to the entire cortical mantle, the olfactory tubercle, and the amygdala. It has been functionally associated with the control of attention and maintenance of arousal. So, it has been seen is that in depression, the levels of acetylcholine are raised. So cholinergic agonists, they produce more lethargy, energy, and psychomotor retardation, whereas cholinergic antagonists in depression have shown to improve the symptoms. So these were the biogenic monoamines. And as we saw that dopamine is basically, it was responsible for the low motivation drive. Serotonin was responsible for libido, sleep, all the circadian rhythms, and norepinephrine is basically known to cause the somatic symptoms in depression. So moving on, let us now study about the monoamine hypothesis. So according to this hypothesis, there is tri-monoamine depletion, which leads to depression, and mania can be due to the opposite, that is due to an excess of monoamine neurotransmission. So this is a very simplistic conventional theory, the chemical imbalance theory. So here is a very simple diagram showing that when there are monoamines present, there is no depression. And here we can see the tri-monoamines are depleted. So this is a state of depression. Now what happened was, as gradually over time, so the focus of the hypothesis for the mood disorders, it basically moved on from just the tri-monoamines to the abnormalities in their receptors, to genes, to epigenetic changes, then to the immunology and endocrinology. So another question which arose was that, since when we give the classic drugs for monoamines, they increase the levels of monoamines almost immediately, then why do the clinical effect is prolonged? Why do the clinical effect takes weeks to occur? So another hypothesis came up, which was the monoamine receptor hypothesis. Now, as per this hypothesis, whenever there is depletion of the neuroamines, there is a compensatory upregulation of the neuroamine receptor. So basically, it was seen that the time taken after giving the drugs for depression, the time taken for the clinical effects to come correlated with the time taken for the down regulation of these receptors. So hence the role of this hypothesis. Next, a very newer hypothesis was the glutamate hypothesis of mood disorders. Now, glutamate, we all know, is an excitatory neurotransmitter and it acts on the NMDA receptors. Also, it cannot cross the blood-brain barrier, so it has to be formed centrally in the neuron and glia. Now, glutamate has both the roles. It has roles in neuroplasticity as well as excitotoxicity. Now, it has been shown that glutamate levels are increased in the plasma levels and in the postmortem samples of frontal cortex from patients with major depressive disorder, and antidepressants restore normal plasma glutamate levels. Now, these glutamate levels can be caused due to the alterations of the glutamate release, the clearance, and the metabolism in selected brain areas. Now, another evidence to support this hypothesis came from a number of clinical studies showing the action of ketamine. Basically, ketamine is a NMDA, N-methyl D-aspartate receptor antagonist, and it is used as an anesthetic agent at doses as high as 2 milligrams per kg. Now, different studies, they reported a robust, rapid, that is within 2 hours, and sustained 1 week antidepressant effect in treatment-resistant patients with major depressive disorder of a single infusion of ketamine at sub-anesthetic doses, that is 0.5 milligrams per kg. However, because ketamine we know has certain psychomimetic effects and dissociative effects, this was not like this impeded a widespread application of this molecule in the treatment of depression. Now, moving on from, we have gone through the neuroanatomy, the neurophysiology of all the neurotransmitters. Then we come to the neuroendocrinology. Now, in neuroendocrinology, we'll be covering the hypothalamus pituitary adrenal axis, the thyroid axis, and then the gonadal axis. So first of all, let us look at the HPA axis. Now, what is the HPA axis? The hypothalamus, basically, it releases corticotropin releasing factor, which acts on the anterior pituitary to release the ACTH, adrenocorticotrophic hormone, which acts on the adrenal glands. So now the adrenal glands we know has adrenal cortex and adrenal medulla. The adrenal medulla releases catecholamines, and the cortex further releases mineralocorticoids, glucocorticoids, and then the sex steroids. So here we'll be talking about the glucocorticoids, which are released from the adrenal gland. So the glucocorticoid cortisol, it is a stress hormone. So it is released in response to stress. So now it has been seen that in mood disorders, there is an HPA axis dysfunction. Also, there is a loss of feedback inhibition to the hippocampus. Now, affective disorders often occur secondary to stress. Now, what was seen was that neurons from the hippocampal area and the amygdala normally suppress the HPA axis. Now, due to stress, due to the release of glucocorticoids, these neurons in the hippocampus and amygdala, they were damaged. Hence, the inhibitory response to the HPA axis was also diminished. So this led to the overactivation of the HPA axis and hence hypercortisolemia, which was seen in mood disorders. Also, as we said, that there is impaired sensitivity to negative feedback in the HPA axis dysregulation. The HPA axis overactivity appears to be particularly enriched in the melancholic and psychotic depression subtypes. Now, the dexamethasone suppression test. Normally, what happens is when we give dexamethasone, it is a very potent glucocorticoid. So whenever any exogenous steroid is administered, it leads to the negative feedback, and hence the endogenous glucocorticoids are not released. But since in the HPA axis dysregulation, which was seen in mood disorders, there was this loss of negative feedback. So even after, in depressed patients, it was seen that even after giving dexamethasone from outside, it at around 11 p.m. it is given at a dose of 1 milligrams. The next morning, the cortisol levels are checked. So it led to the failure of suppression of the cortisol levels next morning. So it is not diagnostics, as it is also seen in dementia and eating disorders, but it has a prognostic value to it, as the non-suppressors have increased severity of depression and mood relapse rates. So to summarize, the HPA axis alterations which are associated with depression include blunted cortisol circadian rhythms with elevated levels late in the day. Late in the day as in because the cortisol follows the circadian rhythm. It is highest at the time when we wake up until and at the time of midnight, it is at its lowest levels. So this was blunted in mood disorders. Negative feedback in sensitivity, as we said in the dexamethasone suppression test, and increased release of cortisol in response to exogenously administered ACTH. blunted ACTH response in response to CRF administration, and increased cerebrospinal fluid CRF concentration. Now let us come to the hypothalamic pituitary thyroid axis. So this is the hypothalamus, this is the axis. From the hypothalamus, there is release of thyrotropin releasing thyroid releasing hormone, which acts on the anterior pituitary to release the thyroid stimulating hormone, and then the thyroid gland, which further releases T4, which is converted into triiodothyronine. Okay, so clinical hypothyroidism is frequently associated with depressive symptoms, and subclinical hypothyroidism, that is defined as elevated serum TSH with normal T3 and T4 levels, is commonly reported in treatment-resistant depression. However, most patients with depression do not have any biochemical evidence of thyroid dysfunction. Now, studies suggest that clinical effects of T3 as an augmentation agent with antidepressants in patients with major depressive disorder who have not responded to antidepressant movement. Now let us read about the supraphysiological doses of levothyroxine which have been given in mood disorder. So supraphysiological doses above 150 mcg micrograms per deciliter. So the supraphysiological doses of levothyroxine has demonstrated efficacy in reducing depressive symptoms, rapid cycling, prophylaxis resistant bipolar patients, and for patients with refractory unipolar or bipolar disorder. Now, despite its efficacy and effectiveness, a broader acceptance of the treatment with supraphysiological doses of levothyroxine has been limited by the sparse number of long-term studies investigating its tolerability and potential somatic side effects. Now, when these supraphysiological doses were given to these mood patients, they became hyperthyroid, or even they were classified as thyreotoxicated. So this limited the prescription by psychiatrists. Also, we know that giving high doses of thyroid hormones has somatic symptoms. It increases the risk of osteoporosis, increases the risk of cardiovascular risks. For example, it causes tachyarrhythmias, atrial fibrillations, and systolic hypertension, especially the high output cardiac failure. Hence, the evidence supporting the administration of levothyroxine in mood disorders it is not prescribed. Now, let us come to the hypothalamic pituitary gonadal axis. In gonadal axis, we'll be studying both the ovarian hormones and the testosterone. So first of all, let us look at the axis. So this is the axis here. We have the hypothalamus, which releases the gonadotropin releasing hormone, acts on the anterior pituitary to release the luteinizing hormone, and the follicle stimulating hormone, which acts on the gonads, both the ovaries and the testes, to release in females the estrogen and progesterone, and in males the testosterone. Now, let us also have a look at the menstrual cycle here. So we know the menstrual cycle is of 28 days. At the 14th day, there is ovulation. So starting from the day 0 till the day 14, there is rise in the follicle stimulating hormone, basically FSH. It acts and it causes the growth of follicles in the ovary, and follicles release estrogen. This we all know. So at the 14th day, there is an LH surge. So when there is an LH surge, there is ovulation, and estrogen levels decline and progesterone levels start to increase. So now during the end of the cycle, just before the in the late luteal phase here, the progesterone as well as the estrogen both keep on declining. So in this phase, it has been seen that there are mood symptoms in girls, like depressive symptoms. So this has been known as the PMDD, the premenstrual dysphoric disorder. So as it was seen that estrogen enhances the release of hormones, and decline in the levels of estrogen basically led to mood symptoms. It is also seen in, for example, in menopausal women and postpartum. Okay. Then came the role of oral contraceptives. Now, oral contraceptives, basically there's a confusion that they have been linked with the potential to alter mood. Now, there is a controversy that whether they worsen mood, improve mood, or they are mood neutral. Now, one study showed that oral contraceptive use is associated with increased risk for subsequent antidepressant treatment, depression diagnosis, and suicidal acts, with significant risk for all hormone preparations, especially the progesterone hormones. Now, hypothalamic pituitary gonadal axis, the testicular hormones. Now, depressive symptoms are common in men referred with borderline testosterone levels, and middle-aged non-depressed men in the community with low testosterone levels have a higher likelihood of developing depression. The bulk of the evidence suggests that testosterone does not have efficacy in treatment-resistant major depression as an adjunct to antidepressants, and its routine use would not be clinically appropriate. Now let us come to BDNF. So BDNF is brain-derived neurotrophic factor, and it is a very important growth factor in brain. It has roles in neurogenesis and neuroplasticity. So stress, inflammation, genetic and environmental influences, it leads to the loss of growth factors, leading to lack of synaptic maintenance, loss of synapses, dendritic arborization, and ultimately leading to loss of neurons themselves. Now, in mood disorders, it has been seen that there is decrease in the level of BDNF in both manic and depressive stages of bipolar disorders. MDD patients also show a remarkable reduction in serum BDNF. Now, in this figure, as we can see, that this is a neuron, and a brain-derived neurotrophic factor leads to a better synaptic maintenance and growth of neurons. But if the genes for the BDNF, they are shut down, it will lead to the apoptosis of neurons. Now let us come to psychoneuroimmunology. Now, mood disorders, they are often associated with chronic low-grade systemic inflammation with increased plasma levels of pro-inflammatory mediators targeting all tissues, including the brain. Importantly, the pro-inflammatory cytokines include the TNF-alpha, interleukin-6, which regulate mood behavior. Now, it has been seen that chronic stress and inflammation, they are driven by contemporary Western lifestyles, including stressful social environments and unhealthy dietary patterns, limited physical activity, and obesity. So basically, what was seen was that prolonged stress, chronic stress, it led to the activation of microglia in the brain, and activation of microglia led to the release of pro-inflammatory cytokines, and hence there was a chronic low-grade systemic inflammation. And these pro-inflammatory cytokines may aggravate short-term mood changes to a chronic maladaptive depressive state by preventing the normalization of mood after adverse life events. So it was also seen that many anti-inflammatory drugs, it was seen, for example, in NSAIDs, it was seen that toricoxib has even antidepressant effects. And also it was seen that post-COVID, in COVID, we knew that there was cytokine storm. So when there was cytokine storm, it was seen that post-COVID depression cases, they were more. So it was basically linked to this hypothesis. So in this figure, there are multiple levels of evidence of immune dysfunction. For example, in the CNS, as we said, increased level of pro-inflammatory cytokines in the CSF, microglia activation in the peripheral blood, there was increased oxidative stress markers, autoantibodies, pro-inflammatory cytokines. Clinically, improvement of mood symptoms with anti-inflammatory drugs was seen. And now, how do these pro-inflammatory cytokines act? They basically, as we said, that BDNF was responsible for neurogenesis and neuroplasticity. So the pro-inflammatory cytokines decreased the production of BDNF. Also, there's an enzyme which is known as tetrahydrobioprotein, which is a coenzyme in the monoamine production. And this was also, its activity was also compromised. Also, the production of serotonin decreased due to the pro-inflammatory cytokines. Now let us come to the circadian clock, the broken circadian clock as seen in the mood disorder. So we humans, we are diurnal organisms. That is, we have a circadian rhythm to follow. Now circadian rhythm is basically a 24-hour biological internal clock. Now circadian rhythm, it does not only include sleep. It includes the basal body temperature, the blood pressure, certain hormones like cortisol, and certain genes which basically act in the 24-hour cycle. So these all comprise the circadian rhythm. Now, circadian, so sleep basically is very important. So brain, basically our brain is 2% of the body mass, and it consumes 20% of the energy output. So for example, like a car engine. So a car engine needs fuel, it consumes fuel, and then it gets heated up. So when it gets heated up in a car, there is a coolant to cool it down. So in our body, we do not have a coolant. Okay, so heart, from the heart, the blood is pumped at 37 degrees Celsius, and the same blood at 37 degrees Celsius goes to the brain and when taken out to the skull circulation. Now, the coolant in our body is sleep. Hence, sleep is very important. Now, it has been seen that from 10 p.m. in the night to 2 a.m., there is reduction of metabolism by almost 44% in the brain, and there is ATP generated in the brain during this time. So, sleep during this time is very important. Now, if we'll not sleep at this time, we'll be suffering from lack of energy in the next day. Okay, so now also sleep basically it was seen that sleep is disturbed in mood disorders. Now we have we have certain environmental cues which synchronize our circadian rhythm. Those are known as Zeitgebers. So for example, light is a Zeitgeber for sleep. So light, it acts on the retinal ganglion cells, then it goes to the suprachiasmatic nucleus, then to the pineal gland to release melatonin. And hence this circadian rhythm is maintained. So it is very important to it is very important. Early morning sun exposure to the blue wavelength of the sunlight is very important. And as the day passes by, the sun goes from yellow, orange, red. That is not important, but the blue wavelength is important. So also numerous genes that operate in a circadian manner sensitive to dark light rhythms are called as clock genes. Now, abnormalities in these clock genes have been linked to mood disorders. For these patients, circadian rhythm treatments such as bright light, melatonin, phase advance, phase delay, and even sleep deprivation can have therapeutic effects. Now, neuroradiology. So, in neuroradiology, the structural changes. So, ventricular enlargement, cortical atrophy, sulcal widening, and especially the reduction in the size of hippocampus. So, in depression, in patients of major depressive disorder, it was seen that the hippocampus can shrink by almost 20%. Also, it was seen the reduction in size of hippocampus was also seen in patients of Alzheimer's. And other structural changes include the increased size of pituitary. The functional PET changes are decrease in anterior brain metabolism, decreased activity in frontal cortical areas, hyperactivity in amygdala, which is the emotional circuit, and increased frequency of abnormal hyperintensities in the subcortical regions, such as the paraventricular region, thalamus, and the basal ganglia. The functional MRI, greater activation in frontal and anterior temporal regions during inhibitory tasks was seen. Now let us come to genetics. Now several twin studies of mood disorders considering unipolar and bipolar disorders together find a 2 to 4-fold increase in concordance rates for mood disorders in monozygotic twins as compared to dizygotic twins. Now, heritability increases in proportion to the amount of shared genetic material. Thus, early age of onset is associated with greater heritability. Further studies show that heritable risk transcends environmental influences. Some of the changes in brain are which are attributable to the chronic stress may be mediated by epigenetic changes. Now, some of the genes which are involved are the serotonin transporter gene, the BDNF, CRF gene, the DRD4, and SLC6A3 gene. Now, this is a list of the selected genes which are associated with major depression and bipolar disorder. These are my references. Thank you. So basically the monoamine hypothesis. So we have basically three monoamines which are implicated in mood disorder: serotonin, dopamine, and norepinephrine. So this is a very simplistic conventional theory. We just said that the decrease in the amount of these monoamines led to symptoms of depression, and vice versa, an increase in the amount of these monoamines led to manic symptoms. This is basically what is called as the chemical imbalance theory. Concept is associated with increased or decreased risk. Basically, it is not very very clearly well defined that whether it is mood neutral, it increases or how it affects the mood. But some studies have shown that the use of oral contraceptives is basically more associated with the increased use of antidepressant in the later course of time, especially the progesterone inhibitors.
So you said that when using anticholinergic drugs would be beneficial. Can you elaborate?
So it was seen that in depression, there was a rise of acetylcholine levels. So the acetylcholine levels they were increased in depression, and so it was seen in some studies that anticholinergic inhibitors, for example, triacetamide, which we give, it can even lead to resolution, to some extent, resolution of the depressive symptoms.
So reference
So it was an article.
journal name because little clinically we don't see this first of all, and then all the antihistaminics, all the anticholinergic drugs, they should actually give us happiness. They give a lot of dysphoria. And then whether it was a human study or an animal study, and how they measured it, all those things become. And you mentioned that one place receptor regulation is involved in happiness. Can you explain? So that's a very important part.
Yes. So basically, what was seen was that when we get classic drugs for depression, so they lead to an increase in the neurotransmitter levels. So when there is, if on giving the drugs, there is an immediate increase of levels of neurotransmitters. So why the clinical effects they come after these, they should come immediately. So this was explained by this hypothesis. So this hypothesis said that when there is a decrease in the levels of neurotransmitters, as shown here, there is an upregulation of these receptors. So it was said that after giving the classical drug for depression, the time which was taken for the clinical effects to come, it was basically correlated with the time taken for the down regulation of these receptors. So this was the hypothesis.
Then how it explains? Because if upregulation is there, less mono means more receptors, it will give more kick to the brain. So then how it works out? Because why receptors levels have to go down in order for these antidepressants to manifest? Because you're saying that all these are required, the brain is adapting. So why the adaptation has to reverse? Why this happens? Yes, autoreceptors. Because autoreceptors have to, there are three sides of autoreceptors, in which basically the function of those autoreceptors are they actually keep a break to the release of these monoamines in the synapse. So when you start antidepressants, actually the down regulation of these autoreceptors starts happening, and time duration of these antidepressants is correlated to the down regulation of these autoreceptors. They're actually pre-synaptic. Subsequently, the break goes off and means it start releasing, and so this is more of. So the concept is fine, but what about second business systems, third signaling, anything in that context? JAK-STAT pathway, it has your TRK receptors, anything in that context. Just touched on BDNF. BDNF is just one of the growth factors. hundreds of those. Unfortunately, BDNF got more attention than actually it should have got. There are actually other good factors. Because now most of the neurobiology has shifted from these hypotheses of means to these areas, to these pathways, to circuits and signaling. And we briefly touched upon those inflammation and psychoneuroimmunology and endocrinology. So I think most of things are now being promoted. So maybe we have to have a version 2 of neurobiology disorders where all these things are being discussed. Ketamine, if it works, then how it works, why why it works so fast, why the mechanism of action is very fast. So all the answers lie in all these areas. The role of protein kinase C, protein kinase A, and depression. These are important. Any questions?
Lot of our drugs
Increase of tension.
so there's no so there are two arms positive and negative. The positive as it is associated with genes clock and PN and the negative is associated with so it has been seen that these genes are also associated with mood disorders. And this is the statement, conflicting statement that was written in the US reports that there is a phase delay in mania and there is a phase advance in depression. But then in another study it was written that there is a phase delay in depression, whereas in mania there is reduced latency of death. So what is written in DSM-5 that there is a decrease that is not actually there. It is that sleep is sustained from sleep.
So what meaning?
Meaning was that phase delays what I could understand was the Vanjik was silly and advance was just Vanjik for 10 years. So in a depression there is early morning awakening and typical.
Melancholy depression, the early morning awakening. Many times
Typical sleep delay.
They will sleep very, very high. So here the sleep restriction is there. So, no, because depression is also very serious. So not have a single template of depression.
In order for this to be treated, the fail advance in depression and typical depression, 4 hours like you have to like
And default mode network, salience network, central executive network, DMN, whether it stays in DMN or the DMN switch off, it becomes a very more type of factor. What is DMN? What are areas? Long before we had these presentations, we can give the interns one network. There are 4, 5 attention networks, salience, central executive, DMN. So DMN, what is DMN? DMN network which connects to the anterior and posterior areas of the brain. What are areas of the brain? So these are very important. So what happens in depression? the so Dr. Sarkar, senior now. Comments?
So, we discussed about competition theory. We discussed about so when we call them bias, they said that women actually use cities from a protective factor that they are educated. But there is also like tendency to exhibit high risk behavior like smoking and they themselves like involving in unprotected sexual intercourse also exhibiting high risk behavior. So to all such research, they have the propensity of developing more depression. So that's why it is said that depression is more. We say that it's like a very biased.
When you say the bias for founding factors. See, if you really want to study effect of OCP in a population, so all these factors can compound the outcome. If it is depression, all these factors can influence the outcome of depression. So it seems that OCP is using population. These are compounding factors which needs to be controlled. You are right. But OCPs is because long before what we were reading that low estrogen expectations were less associated with new psychiatric sequelae. You are saying the estrogen is required to release but it is not very simple. It's very conflicting information is there when we talk about reproductive system and your depression or psychiatric sequelae. So there is, you can't simplify things. We need to dig it out. Dr. Ishani can take it up in next seminar, next series. Reproductive system and depression for some real female substitute. So, Ayushi, Sarthak, points. कल अगली बार से पढ़ के आना है सीरियस आता है आपकी और सेकंड ईयर में आना है
Circadian rhythm that is regulated by regulated by our these clock genes in human. So the clock gene is what it does. It is clock gene, clock BMR, they go and bind the key box side of the computer within it's not so this clock gene is protein of they go
I think what a good topic for this. Clock genes. Why not officially take it? Or you take it as the reproduction part of it and you take it as the slot. Yeah, it's more than the next March. Maybe a presentation kind of a handouts that will be something daily. And this is a little interesting. Clock genes are a lot of work. Okay, Dr. Nidhi, very the new group results are very bright, so I think we have 3 intelligent people going out and 3 intelligent people coming in. Dr. Anusha, neurobiology sort of a huge topic. You will encounter this in the next 3 years, either in your academics, bedside, treatments, viva, theory. It is very important. Maybe say everyone of us develop depression despite having the risk factors. It's the only reason being that we all adapt. Once you surpass that strict drive, it can be found out of many reasons. One of the reasons are harm. So abnormal is not because किसकी वजह से ही होगा. There are various other reasons, inflammation, inflammatory disturbs, and your retinal system is there, and immunity is there. There are a lot of issues, but this is the reason why, as she said, that post-pregnancy, the problem happens, or it happens in post-menopausal phase, or in narky, or for motor psychiatrists, for example, in female psychiatry, or in these two cases because of the similar reasons. Increase or decrease, we can later on understand, but there are because of fluctuations. That is why you will see that in pregnancy things are not very simple. You will see there is a slow and serious rise of estradiol or progesterone. So actually, few disorders, right? Few disorders, late, and like in migraine, 130 degrees, 130 degrees, 130 degrees. And similarly with depression, but there is no any consensus reached because of the lack of clarity in our understanding. So maybe we can have a presentation. Your doubt is clear. So पहले मैं बताता हूँ फिर आप as a chair after last meeting. I think very good presentation. We tried a little bit because point is it's a very difficult center, very huge center. Still we are not able to go a few areas. Maybe later on we can specify what all areas to cover. But I think it was a very good attempt to present because each and every subsection are very difficult to remember and subsequently deliver those. I think very content is very rich. So I hope this would have come in a handout, maybe which we will be planning subsequently. I think it's a good presentation. So आप जब लोग सेमिनार हैंडआउट्स बनाएंगे ना तो हम उसके बुकलेट बना लेंगे. वो बुकलेट हम लोग बाहर भी रिलीज करते हैं. आप लोगों का नाम कितना फेमस हो जाए. सेमिनार में जैसे प्रोफेसर आकर के नाम भी नहीं लिखता, डिपार्टमेंट का भी नाम नहीं लिखता, वेन्यू भी नहीं लिखता, डेट भी नहीं लिखता. So you have to write everything. What you can do is from Srinivasan you can have a seminar. Here's a list. You can see how they meet.